Okay, good afternoon. Could you maybe please take a seat? We're running a little bit behind schedule. I'm looking very much. My name is Bart Eggen. I'm chairing this next session, which is about Alzheimer's and dementias. And excited, good lineup. So the first speaker is Michael Heneka from Luxembourg. After seminal work on the inflammasome, now he has found the tunneling nanotubes, which is a fantastic story. So I'm looking forward to it, Michael.
Thanks, Bart. I want first to thank the organizers, and especially Guy, for inviting me and giving me the opportunity to discuss this new and completely unpublished set of data with you. So, as we all know, Alzheimer's disease is characterized, as you can see here, in this Biochemical stain by extracellular intraneuronal accumulation of proteins. By now we know that this is tau, hyperphosphorylated tau, extracellular deposits made up of amyloid beta. These have been characterized. But what some of you may not know, that Franz Nissl and Alois Alzheimer already depicted the suffering of microglia cells. And in the first publication, there are approximately 1.5 pages about the extracellular deposits and neurofibrillary tangles. And then there is 7 pages of microglia pictures. So I think they did not underestimate the role of neuroinflammation and microglial changes in this disease. And by now, we know through epidemiology, clinical observations, genetics, experimental models, biomarkers, that inflammation has a major impact on this disease. But as in Alzheimer's, there are other diseases which include Parkinson's and Lewy body disease, to some extent AD as well, which are showing further intracellular protein accumulations, in this case, Lewy bodies made of alpha-synuclein. And we had this very interesting observation some years ago, which taught us that microglia cells would on demand form a network to share their cargo burden and would distribute overeaten alpha-synuclein aggregates with each other in order to survive. And one part of the survival was that the microglia cells, which would have phagocytosed too much alpha-synuclein aggregates, they would receive mitochondria from incoming microglia cells connecting via structures we call tunneling nanotubes. And when Hannah and Frederick came with these data to me, I thought, wow, that's pretty interesting. It's cool, I haven't seen that before. But I immediately remembered one painting by Alois Alzheimer himself, another one. This is what he called amoeboid glial cells. It's a microglia cell, and he pictured them in various positions close to neurons, and this one showed aggregates inside. And so I had the idea it would be cool if microglia cells probably would hook up with neurons as well and help them to get rid of these aggregates. So in fact, they do. This is various an immunohistochemistry, which shows you structures that connect the neuron with the microglia cells down here. I hope you can see that these cells are really connected. This is hopefully easy to see. What are tunneling nanotubes? Tunneling nanotubes are open-ended, direct connections up to 100 micrometer of length, 20 to 700 nanometers width. And they are filled with cargoes that are being shifted along cytoskeletal filaments. And a frequent cargo, for example, is mitochondria. So glioblastoma, in fact, fuel their energy need by getting mitochondria from invading macrophages through tunneling nanotubes. They exist as single roads, but also as highways. There are various proforms which are on the right side here, and once these protrusions from the microglia cells have invaginated the neighboring cell, there is fusion of the cell membranes, and then these open endings exist. So what do they do? This is another example. This is another electron microscopy, and here you can see neurons, microglia cells connected with each other. You can see these long, free-floating tunneling nanotubes. To some people in the TNT world, it's important that they are really floating, that they are not touching the ground. They do so when they transport heavy cargo whatsoever. This is directly a position of fusion. Now, one important thing is, in cell culture dishes, you can observe a lot of artifacts. Is that also something that occurs in real life? And this is a data set from the Microcons Consortium, who provides serial sections of the mouse cerebral cortex on an EM level to the public. And here we identified several positions where there is no membrane actually between, as you can see indicated with the white arrows, no membrane between the microglia process and the neuron process. So they touch each other, and the question is, what does that mean and what it is good for? Another picture, and I would want you to watch now the little movie. This just proves that we are, if you see here, this thing coming up. So this is a Z stack. It's really hanging inside. There's another one. It's really hanging in the, not touching the cellular stratum, so the bottom of the plate. It's connecting these cells in these long, thin tubes. Now, this is another picture just to give you an idea. Distance to the substratum, the microglia cells have more volume, the neurons are more flat, so there is this little kind of not even leveled phenomenon. You see still that they're really long and thin structures. There are about 2 to 6 per microglia cells connecting to a single neuron, and their length is around 10 to 15 micrometers in our cell culture condition. Now, if we expose neurons, or if we increase alpha-synuclein aggregates in neurons, or we induce that aggregation, or we add aggregates and they're taken up by neurons, and we add in microglia cells, there is an increase in the number of tunneling nanotubes formed by microglia cells in response to these signal from neurons, but they don't change in morphology. So length is equal on all the other features as well. But what do? So they're quite good in actually taking out the aggregates from the neurons. And you will see now various cells, various microglia cells added into this neuronal culture. You see the fluorescent alpha-synuclein is restricted to the neuron here. And if we go on, you see that there's one coming in, forming a tunneling nanotube. There's another one who forms 3. And over time, they take up the aggregates and they free the neurons from the aggregate. We can do this in time-lapse analysis, and then you can follow single aggregates that go from the neuride or from the axon into the microglia cells, and actually we also can visualize degradation within the microglia cells. So this is an experiment with alpha-synuclein. This is another experiment you see here at various positions. I hope you can, this is not my favorite way of showing it. You see, and I'll try this. You see these cells here, they're empty. In red, you see these 2 tau overloading neurons, and if we proceed in time, you will see how these microglia cells, which are added in, take up, and how the neuron is freed from the aggregated tau. So this is the dynamics of it. We followed this up to 6 hours, And you see, regardless whether we look to alpha-synuclein or tau, the microglia cells take up the aggregate in a time-dependent fashion. But as at the moment they take it up, there's also already degradation setting in. And I don't show this, but if we would stop here after 24 hours, the microglia cells would have degraded the entire cargo. Now, you could argue that this is due to phagocytosis of neuronal structures. And we proved that, so neurons were cell tracer labeled, and you see indeed there's a little uptake of neuronal structures at the beginning, but then there we can clearly distinguish the uptake of alpha-synuclein or tau independently from cell tracer. We had another experiment to prove that it's really tunneling nanotube dependent. Here you see that we use an insert control, and when microglia cells are added into cell culture, we have a decrease of the mean alpha-synuclein fluorescence in the neurons. If we do that in a microglial insert, then this is not the case. So this proves to us that there has to be a physical contact. When we challenge neurons with alpha-synuclein, we see profound transcriptomal changes on the neuronal side. There are various pathways upregulated contributing to ribosome biology, oxidative phosphorylation, Parkinson's, Alzheimer's, and Huntington's, so you have them all here. At the same time, a lot of homeostatic pathways are being downregulated, and I want you to keep that in mind. Now, when alpha-synuclein and tau are actually accumulating in neurons, there's an impairment of mitochondrial function in these neurons. These are seahorse experiments, and you can easily see that there is a change in basal and stimulated respiration. And it's even more obvious, this breakdown here in response to tau. So having this kind of metabolic or respiratory problem led us to ask the question whether, like in the microglia cell culture, microglia would also be able to deliver fresh mitochondria to the neurons. And indeed they do. So these are two microglia cells which have been popped on a neuronal cell culture. And as time proceeds, you will see how they distribute their mitochondria into the neuronal network so that it becomes visible. And this is easy to quantify. We see that the number of mitochondria, labeled mitochondria, decreases over time in neurons. At the same time, they increase in the mitochondria. They integrate very well in the existing neuronal mitochondria network. If we look at a functional level, we used the TMRM assay. You see that adding alpha-synuclein or letting alpha-synuclein being overexpressed by neurons, you have this loss of this mitochondrial membrane potential. If we add in microglia cells, this is rescued, as is the mitochondrial elongation score. You also can see that this, obviously, it's easier to see here in the quantification in the bar graph, like alpha-synuclein decreases it, and adding in microglia cells would rescue this effect. Now, the question is, does that have anything to do with neuronal function and survival? How we can prove that these tunneling nanotube formation and this transfer from cargo as well as delivery of mitochondria has any impact? So we set out to look at basal neuronal network activity through calcium transient imaging after GCAM6F transfection of neurons. You can see this is how it looks under the microscope. We look to frequency, we look to amplitude, and half-width as parameters of analyzing this basal network activity. And one thing is really easy and very reproducible to see under controlled conditions. You have these frequency here, burst frequency. This is being silenced as at the moment that neurons are being loaded with alpha-synuclein. But if we add in wild-type microglia, we not completely, but very much so restore that frequency. And this is the quantification of that. It looks, well, this is an experiment where we are not no electrophysiologists, so I was very critical with it. And this is actually an experiment done by Kai Jüngling and Hans-Christian Paper from the University of Münster in collaboration. And they're able to patch clamp single neurons and analyze them. So what they did is they looked at these neuronal burst rates, added in alpha-synuclein aggregates into the neuron, again silencing this, and then adding primary microglia cells. And as you see, after 6 hours, it's not entirely restored, but it's very much back to normal. So in the same time, there are two things happening. The microglia, obviously, which surround this neuron, they take up the aggregates, but they also deliver fresh mitochondria. Now, same thing with tau. If we do this experiment with tau, burst frequency is silenced with tau, adding in primary microglia and allowing them to transfer the aggregates out of the neuron is very helpful and restores the original frequency pattern of the basal neuronal network activity. And this is an experiment where, again, we looked at whether it would work equally well, and it does not. There is no rescue at all when we use the transwell experiment, kind of proving that we need this direct physical contact and giving the cells the chance to connect via tunneling nanotubes. So there are obviously no soluble factors involved in this phenomenon. Now, what about survival? When we look to the generation of radical oxygen species, we see that alpha-synuclein load of neurons increases ROS levels. And it's very easy to see that after 1 hour of adding in microglia cells, there is no effect. If we let the microglia cells do their work for 6 hours, there is a significant reduction here. In the insert well experiment, this doesn't work at all. Cytotoxicity, a measure of neuronal viability, shows you that alpha-synuclein load lowers or increases neuronal cell death, and there is no effect after 1, but a very strong, almost complete rescue after 6 hours of allowing microglia cells to help neurons via tunneling nanotubes. Now, this looks pretty much the same when we look to tau. We just wanted to prove that this is not an alpha-syn phenomenon. This is something which is a more general principle. You see tau is increasing ROS levels and increasing neuronal cell death quite a bit. And again, adding in microglia cells almost completely rescue this phenotype, while having the insert well experiment, which does not allow for TNT formation, would completely prevent that. So we know that we have to show really what is the contribution of the mitochondrial transfer. And what we did in this experiment, we intoxicated microglial mitochondria with treating them with antimycin A, which is an irreversible complex 3 blocker in mitochondria. And if we do this, we have no effect. So this is the ROS level experiment. The Cytox experiment looks identical. You see, under normal conditions, if we allow microglia to provide healthy mitochondria, there is a complete rescue of ROS levels in the alpha-synuclein as well as in the tau experiment. If we use antimycin A intoxicated mitochondria, in both cases, there is absolutely no effect. So that kind of proves that the microglia are really capable of delivering mitochondria to the neurons via tunneling nanotubes, which rescue in that cell culture experiment, that type of alpha-synuclein or tau-induced neuronal cell death. So, but that's not all. I think I showed you which of the pathways were up in response to alpha-synuclein challenge within neurons. And interestingly, if we allow these microglia cells to form tunneling nanotubes and do their job, then all these pathways which have been previously upregulated, Alzheimer's disease, Parkinson's disease, and Huntington's disease, are downregulated, while a number of homeostatic pathways are coming back up again. So there is not only this effect on the survival, on the ROS function, or on the network function. There's a really direct effect on the transcriptome of the neurons. Now, what about mutations? I told you now a couple of times we worked with alpha-synuclein with a focus on Parkinson's and LBD, and we worked on tau, which is more related to Alzheimer's disease and frontotemporal dementia. So we looked at microglia cells which were having a mutant for LARC G2019S. As most of you may know, this is the most frequent LARC2 mutation. And indeed, the LARC mutant microglia did not show the same type of efficacy in transferring out alpha-synuclein aggregates from neurons. They even tried to make up for it by delivering more mitochondria, but we know that LARC2 mutant cells have a defect in mitochondrial respiration, and overall there was no rescue of the ROS levels or the viability of the neurons. We also, this is just showing you the experiment proving that the LARC mutant has an impaired respiration. So that's probably the reason why they try to deliver more and more mitochondria into neurons, but because they're defective, they're not helping. This is an experiment showing you that we used iPSC-derived neurons and microglia cells from a mutant carrier and controls, which show that we basically observe the very same phenotype in human iPSC-derived cells. So it's not a mouse phenomenon either. TREM2, of course, our favorite kid on the block since a couple of years with respect to AD genetics. TREM2 T66M, however, is associated with frontotemporal dementia, autopathy. And for that reason, we used to study it in the tau model. Here you see that T66M mutant microglia are less capable, like by about 40 to 50% reduction of transfer of tau out of the neurons. There's no change of the mitochondrial delivery, and overall, this does not result in any rescue. Now, watch this. Sometimes nature invents things a couple of times. This is out of the internet. You will see that all the other cells come in. There's a signal, obviously. There's a signal calling for help. And they don't stop, actually, until they've turned the ship around. So in summary, I tried to show you that microglia cells form tunneling nanotubes with neurons. I find this really spectacular because it's clear that, microglia with microglia, maybe endothelial cells with endothelial cells can do that, but these are different cell types. And this happens in cell culture, in acute slice cultures, in animal models, and what I didn't show you, also in human brain. TNTs allow for the transfer of alpha-synuclein and tau aggregates from neurons to microglia. At the same time, there is exchange of mitochondria, rescues neurons from oxidative phosphorylation stress, increased ROS levels, and cell death. And ultimately, microglia protect neurons from dysfunction through TNT-mediated support. And this involves both sides of the coin, the transfer of the aggregates as well as delivery of mitochondria. And together we suggest that this could be a potential mechanism by which microglia serve neurons in a very direct way and protect them from aggregate dysfunction and death. And let's go to the last and most important slide. Of course, only the person talking about things. These are the people who have done most of the work. Was really spearheaded by Hannah and Frederick, but in collaboration with all these fantastic scientists. And of course, we're thankful for these institutions for providing us with money for research. Thank you for your attention.
Okay. Many thanks, Michael. Wonderful story. It shows that TNT is explosive in many aspects. questions? Yes, over here.
Yeah, amazing work, Michael. I'm really interested to know, obviously, the TNTs maybe increase their ability to clear. Is there a molecular sensor, or what's the sensor that's doing this, or have you worked that out yet?
Yeah. So in some parts of it are a raw signal from the neurons and can be scavenged away with PTIO or with other scavengers. It's not 100% effect, so I wonder what else of signal is there. It's a receptor mediated, so the tunneling nanotubes do not go by chance to the cell. I would not want to disclose which receptor is responsible because we try to protect that at the moment. We believe it can be harnessed for a probable therapeutic intervention. The molecular mechanism behind it involves a limb kinase step, and we have detailed the entire pathway for both the transfer as well as the mitochondrial delivery.
Hi, Michael, question here. It is a very nice talk and really inspiring. Since microglia can form the tunneling nanotube with neurons, so my first question is how microglia can recognize neuron and form this structure? And the second one is whether microglia can form this nanotube with other cell types.
Yes, that's two good questions. One, again, is, as I tried to explain before, it's like a raw signal coming from the neurons, which obviously signal trouble to the microglia cells and start to increase the number of tunneling nanotubes. And I showed you this one assay where the alpha-synuclein-loading neurons have increased the number of tunneling nanotubes being formed by microglia cells. If we put a raw scavenger, we almost suppress this completely. The increase is not completely away, so I believe there's a second or a third signal. The other question is, I believe that, I know that there is a connection or possible tunneling nanotube formation with astrocytes. We're currently testing with which other cells this is possible in the brain. But I think we have, for each connection, we also have to show that it really happens in the models under in vivo conditions and, if possible, in the brain as well.
Yeah, thank you.
There's a question at the top, and there's one down here.
That was fantastic, thank you. I just wanted to ask you if my impression is right. I got the impression that the nanotubules make contact with processes on neurons, and your schematic suggested it was an axon, and I'm wondering, does that mean that the whole thing relies on good anterograde or retrograde signaling, which might, transport, which might be affected in neurons that are a little bit sick?
That's possible. Actually, I claimed the same when I talked to my students, but they came back with data showing that the TNTs are almost equally distributed over the dendrites, the soma, and the axon. So maybe that impression came from some of the pictures, and then there's always a bias if you show pictures, but the quantification proved me wrong, and so the answer is the distribution is pretty equal. What my question is are certain sites are some sites only for mitochondrial delivery and some for kind of excretion of cargo, and we don't know this yet.
Yeah, here, yes.
Thanks for the really nice talk. I'm interested in the transfer of the tau aggregates to the microglia. Is there an associated inflammatory effect in the microglia? These recently reported pathways via CGAS and PQBP1 in the microglial cytosol.
Yes, so I didn't show that, but by now we did the transcriptome in the microglia cells as well, and there is no inflammatory reaction. What I believe is that the microglia cells fine-tunes what is being taken up and what is being kind of degraded over time. As long as there is this balance between uptake and degradation of aggregates, irregardless whether this is A-beta, tau, alpha-synuclein, as long as this will not result into in an inflammatory reaction. The moment this gets out of balance and you get lysosomal disruption, which is, for example, one of the canonical pathways, I'm sure David has alluded to that, for inflammasome activation, then you get all the inflammatory cascades unfolded, and then things get out of control. What I think is this is more like a system that is trying to keep balance as long as possible. It's interesting to see that you can challenge these cells with, you can drive them into senescence, or you can challenge them with LPS or other stimuli, and then they're less capable of doing so.
Question over here. Yeah.
Fantastic stories, Michael. Thank you very much. Just a quick question on the bidirectionality of the transport, and also are the tubes the same? So mitochondrial transport and transport of aggregates. Does it happen in the same tube? Or
I would have loved to show you a video where the cargo comes from one side, the mitochondria goes into the other direction. No, we never saw that. There are the same tunneling nanotubes used for both things, but not simultaneously. I'm not sure if that's possible. I would have loved to see that, but this was just wishful thinking.
So do you sometimes see a second layer of microglia connecting to the inner circle that if they're overloaded to go to that you have another layer of protective microglia?
Yes, we do. I didn't show that because that confuses with the first paper, and I kept it out. But one of the videos has a microglia cell after the microglia cell, so it takes the cargo on and is even more effective in degrading. But that's a single observation, so I'm usually, I don't want to think about it.
Yeah. So it's there's a lot of this. So last question, short one, because otherwise we're running late. Yeah.
Incredible work, thanks a lot. So just a quick question. You mentioned about healthy microglia will donate the mitochondria to neuron. So what's the fate of this microglia? Will we generate some new mitochondria and do the job?
Very, very good questions. One of the worries we had when we looked for longer time periods would the microglia drain out of mitochondria, but we don't see this. And microglia, neither the microglia transcriptome nor microglia survival is being affected.
Right. Will you be around rest of the day?
Yeah.
So all the burning issues. Yeah. All right. Many thanks.